Okay, students, in the previous class just stopped the genetic code and the mutation. And when I was explaining the insertion of bases, at that time the last what that written is the RAM has big red cap. You can once again see the last 5 minutes of the previous class, and then I have continued here. RAM has big red cap. See, in the first what we have taken the base sequence image, there is no BIG ಇಲ್ಲ ಇಲ್ಲಿ. ನಾನು ಯಾವಾಗ ಒಂದೊಂದನೇ ಎಡಿಷನ್ ಮಾಡ್ತಾ ಹೋದ್ ನಾವು ಬಿ ಎಡಿಷನ್ ಮಾಡಿದೆ, ನಂತರ ಐ ಮಾಡಿದೆ, ನಂತರ ಜಿ ಮಾಡಿದೆ, ಆವಾಗ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಹೊಸ ಕ್ಯಾರೆಕ್ಟರ್ ಅಪಿಯರ್ ಆಯಿತು, and that new character is what? The big one. So RAM has a big red cap. Earlier it was only RAM has a red cap. So means when the insertion of the bases, how the change that taken place in the what? Here what is in any what the strand, any mRNA strand. ಹಾಗಾಗಿ ಅದು ಎಡಿಷನ್ ಆಗಲಿ ಡಿಲೀಷನ್ ಆಗಲಿ changes ಆಗ್ತಾ ಹೋಗ್ತದೆ, and that is maybe also responsible for the mutation and mutation for the food for evolution, that also you know. See I once again take another character here. RAM has another character. RAM has a red cap. Same one I will take. So RAM has a red cap. Now in this here example, what I do is I want to delete the what one by one what here the alphabet one by one. So what I do is see RAM has a red cap ಇದನ್ನ ಹೇಗೆ ಬರೆದು ನೋಡಿ. ಈಗ RAM HAS. Now I remove R ED CAP. I removed what? R and remove ಮಾಡಿದ್ದೇನೆ. Okay. So next again RAM HAS. Now once again I remove E. Now DCA and then what? Here only P. Correct. And then last RAM has I remove also what D, and here the RAM has what? CAP. So RAM has cap. So means what removed? The red what a character removed. So like this here insertion or what deletion, and that result in here change in what the DNA sequence or on the mRNA, and that result in the mutation. So ಹಾಗಾಗಿ genetic code and the mutation. What is this genetic code? So genetic code always written in the triplet and the mRNA strand depicted in the triplet code. ಇದು ಒಂದು ಕೋಡ್ ಅಂತ ತಗೊಳ್ಬೇಕು ನಾವು. RAM is one code. This is a genetic code. See similarly here AUC. This is ARUA and CM like this. So this is one triplet codon, and in this triplet codon any one base sequence changed or complete whatever the triplet codon change, what happened? One base sequence, only one base change, that is what is happened in the previous class you studied. It is a point mutation, and for that best example is sickle cell anemia. No? So like this here in what genetic code, one the complete code change ಆದರೂ ಕೂಡ ಒಂದು character change ಆಗ್ತದೆ, and that also alter the what here the sequence, and that is also the mutation. So this is about the mutation you studied, and with this you are going to study the structure of tRNA structure of tRNA. You know tRNA, it is called the transfer RNA, or it is also called as adapter RNA. This is also called adapter RNA, and this adapter RNA or adapter molecule play very important role in transportation of two important molecules. That is, one is anticodon and another one is amino acid during protein synthesis. That is why it is called a tRNA. The structure of tRNA you studied is the clover leaf like structure. Clover leaf like structure you usually study ಮಾಡಿದ್ದೀರಾ. So I am not giving again detailed diagram of this. And the clover leaf like structure is having a like this shaped, and this is a anticodon binding site. anticodon binding site, and this one is amino acid binding site. The two ends are main, and one is a T loop, another is a D loop, and that all you studied in the online class. And again, I am not drawing the diagram here. Please go through this. Only structure you have to study, and maybe it is asking the 2 marks question. Sketch and label the structure of the tRNA, and that you have to draw for the 2 marks question. So now, so this is tRNA structure you study. Then next you are going to study the main important is the second phase of this one is translation. Transcription over. See, during gene expression or protein synthesis, 2 processes take place. I said the first process is transcription, second process is translation. What is transcription? You already studied, and that chapter is complete. Means that main what portion is completed. Transcription is a process of formation of RNA from the DNA. Now, and that process always taken place in the one nucleus only, that you must remember. So, during transcription, mRNA is also formed, rRNA is also formed, tRNA is also formed. And then the main enzyme involved in this transcription is DNA dependent RNA polymerase. And DNA dependent RNA polymerase 1, 2, 3, that also you studied. And just go through the last class the video. So thereby you can understand that, and also you can go through the book also. Anyhow, now translation is the second phase after the what? Transcription. Transcription ಆದಮೇಲೆ translation ಆಗೋದು. What is translation? Translation is a process of polymerization of amino acid. Translation is a process of polymerization of amino acid into polypeptide. ಅಂದರೆ ಅರ್ಥ ಏನಕ್ಕೆ ಹೇಳಿದ್ರೆ ಇಲ್ಲಿ ಎಲ್ಲ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಅನ್ನು ಒಂದು ಕಡೆ ಕೂಡಿಸಿ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಫಾರ್ಮ್ ಆಗೋದು. ಪಾಲಿಪೆಪ್ಟೈಡ್ ಅಂತ ಹೇಳಿದ್ರೆ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಮೀನ್ಸ್ ಇಟ್ ಈಸ್ ಪ್ರೋಟೀನ್. ಪಾಲಿಪೆಪ್ಟೈಡ್ ಮೀನ್ಸ್ ಇಟ್ ಈಸ್ ವಾಟ್? ಪ್ರೋಟೀನ್. ಇಟ್ ಇಸ್ ಅ ಪ್ರೊಸೆಸ್ ಆಫ್ ಫಾರ್ಮೇಷನ್ ಅಥವಾ ಪ್ರೊಸೆಸ್ ಆಫ್ ಪಾಲಿಮರೈಸೇಷನ್ ಆಫ್ amino acids into polypeptide, or in the other word process of polymerization of amino acid into protein. ಅಂದರೆ ಎಲ್ಲ ಅಮೈನೋ ಆಸಿಡ್ ಒಂದು ಕಡೆಗೆ ಕೂಡಿಬಿಟ್ರೆ ಪ್ರೋಟೀನ್ ತಯಾರಾಗ್ತದೆ, and that is the protein synthesis. Okay, so I think you understood the translation. Translation ಅಂದರೆ ಏನೂ ಅಲ್ಲ, ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಎಲ್ಲ ಒಂದು ಕಡೆ ಕೂಡಿಸಿ ಪ್ರೋಟೀನ್ ಮಾಲಿಕ್ಯೂಲ್ ತಯಾರಾಗುವಂಥ ಪ್ರೊಸೆಸ್ಸೇ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಪ್ರೊಸೆಸ್. And you see here the process of transcription. This is one cell. Just imagine this is a cell, and in this is one cell, and this is a plasma membrane. This is a plasma membrane, or it is also called a cell membrane. And here is a nucleus, and this is a nucleus. No, and in the nucleus there is what is the DNA is there. Nucleus DNA is there, and this is cytoplasm. Okay, see, already you studied transcription, and during the transcription what is taking place here? During the transcription here what? The mRNA formation or the RNA formation taking place, and that is the process is called transcription. What is transcription? And that transcription main unit you studied transcription unit, and as a result here one of this act as a template, and this template is converted into mRNA. ಆ ಟ್ಯಾಬ್ಲೆಟ್ಗೆ mRNA ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಬೇಕು. ನಾವು mRNA ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಿದೆ, ಆ್ಯಂಡ್ ದಿಸ್ mRNA ನಾವು ಎಸ್ಕೇಪ್ ಫ್ರಮ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್. ಈಗ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಂದ ಹೊರಗಡೆ ಬರಬೇಕು. ಎಸ್ಕೇಪ್ ಫ್ರಮ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್ ಆ್ಯಂಡ್ ಡಿಫ್ಯೂಸ್ ಇನ್ ಟು ದ ಸೈಟೋಪ್ಲಾಸಮ್. ಇಲ್ಲಿ ಬಂದ ಕೂಡಲೇ ದೊಡ್ಡದಾಗೋದಿಲ್ಲ. ಐ ಹ್ಯಾವ್ ಮೇಡ್ ಇಟ್ ಬಿಗ್ so that I can show you clearly the picture of the mRNA. Same ಇದೆ ಅದು mRNA. Same one is what here I have made big one, so that I can show you clearly what is a translation. Translation ಏನು ಅಂತ. ಸೊ ಹಾಗೆ ಇಲ್ಲಿ ಬಂದು ಕೂಡ ದೊಡ್ಡದಾಗ್ತದೆ ತಿಳ್ಕೊಬೇಡಿ. ಸೊ I have just made it big. See, here in what? Here the different type of the bases are there. A, U, G. It may be C, C, then U, A, C, then A, U, C, U, 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 A, A, U, and then U, A, G. So these are the what the different the bases that is present on mRNA. This is mRNA. Transcription ಆಗಿಬಿಟ್ಟಿದೆ, mRNA ಬಂದಿದೆ. Now start the process called the translation. Translation taken place by 3 main process. That is, one is called as chain initiation. First one is called as chain initiation. second one is called as chain elongation, and last one is called as chain termination. These are the three main processes that is taken place during the what? Translation. What is chain initiation? Chain initiation always have taken place by what? The initiator codon. Which is initiator codon? AUG is initiator codon, you know. So when what? The chain initiation ಆಗಬೇಕಂದ್ರೆ ಅಲ್ಲಿ AUG ಇರಲಿಕ್ಕೆ ಬೇಕು mRNA strand ಅಲ್ಲಿ, and that AUG is only the initiator codon. It is also called the start codon. Okay. Now during the chain initiation, amino acylation process taken place. So amino acylation process taken place in such a way that here DNA produces tRNA, mRNA produce ಆಯ್ತು. ಈಗ ನೆಕ್ಸ್ಟ್ tRNA produce ಮಾಡ್ತದೆ, and that tRNA already I told you that having a two end. One end it what? Having an anticodon end, and another end it has a what? Amino acid binding end. ಹಾಗೀಗ ಈಗ tRNA from the DNA ಬರಬೇಕಾಗಿದೆ from the nucleus, and it load the anticodon, and that anticodon correctly fit to the codons of the what? mRNA. mRNAದಲ್ಲಿ ಇರೋದು codon and tRNA ತರೋದು anticodon. You must understand properly. tRNA bring anticodon, and that properly fix or complementary to the what? Codons of the what? mRNA. So ಹಾಗಾಗಿ tRNA ನೋಡಿ, so this is the one tRNA produce. ಈಗ ಒಂದು tRNA ಇಂದ ಹೊರಗೆ ಬರ್ತದೆ, and this tRNA what? Bring appropriate anticodon. ಯಾವುದು appropriate anticodon? AUGC ಅದು anticodon tRNA ತರೋದು. So when the tRNA coming out of the nucleus and in the cytoplasm, it search here what? Amino acid. ಈಗ AUG ಗೆ ಯಾವುದು ಅಮೈನೋ ಆಸಿಡ್ ತರಬೇಕು? ದಟ್ ಇಸ್ ಆಲ್ರೆಡಿ ಯು ಸ್ಟಡಿ. AUG ಗೆ ಅಮೈನೋ ಆಸಿಡ್ ಯಾವುದು? ಮಿಥಾಯೋನಿನ್. ಇಲ್ಲಿ ಸೊ ಅಮೈನೋ ಆಸಿಡ್ ನಂಬರ್ 1, ಅಮೈನೋ ಆಸಿಡ್ ನಂಬರ್ 2, ಅಮೈನೋ ಆಸಿಡ್ ನಂಬರ್ 3, ಅಮೈನೋ ಆಸಿಡ್ ನಂಬರ್ 4. Here large number of amino acid that is present in the cytoplasm, that is what? Supplied by us only during our eating process. ನಾವು ಏನು ತಿಂತೇವೆ ಪ್ರೋಟೀನ್, that is protein is digested in the intestine into amino acid. That same amino acid is joined to our what? Cytoplasm. Cytoplasm ಇರುತ್ತದೆ, and same amino acid is used for the synthesis of protein in the cell. That is what you call as the protein synthesis. So that is why here tRNA so bring the anticodon, and then it also has to take the what? Methionine. ಹೀಗಾಗಿ ಸೊ ಮಿಥಾಯೋನಿನ್ ಈಗ this type of connection take place. Yes, this is an anticodon. See, UAC is anticodon. This is structure of tRNA, and then it what here bring the methionine, MET, right? MET. ನೀವು MET ಬರೆದಿದ್ರೆ ಅದು ಏನದು? ಮಿಥಾಯೋನಿನ್ ಆಗ್ತದೆ. MET ಬರೆದಿದ್ರೆ ಮಿಥಾಯೋನಿನ್. So like this, here one tRNA is over. Okay, then the DNA from the nucleus produce one more tRNA, and that one more tRNA bring appropriate anticodon to the what? CCC. CCC ಗೆ anticodon ಯಾವುದು? And it is GGG. See, now for this GGG, so which anticodon you have to write? So that is glycine, GLY. ಅಲ್ಲಿ ನಿಮಗೆ genetic code ಕೊಟ್ಟಿದ್ದಲ್ಲಿ ಇದೆ ಅಲ್ಲ information. Glycine, see, CCC ಗೆ so what? Sorry, not it is a GG, it is a glycine alone. It is for the CCC, and for the CCC arginine, ARG. Not the glycine, it is the ARG. CCC is the what? The codon, and to this arginine it bring what? Arginine. ಈಗ ನೋಡಿ ಏನಾಯ್ತು ಅಂದ್ರೆ methionine and arginine, that is what linked with one another, and this bond is called as what? The peptide bond. This bond is called as a peptide bond. Means methionine and arginine are the two amino acid linked with the bond called the peptide bond. Similarly, DNA produce one more tRNA, another tRNA produce ಮಾಡುತ್ತೆ, and that one more tRNA, that anticodon is what here? A, then U, and then G. ನೋಡಿ, so AG. UAC ಗೆ ಯಾವುದು ಬೇಕು? ಈಗ is again one more link is taken place, peptide link, and then dipeptide bond. One is bond, here is bond formed, dipeptide bond is formed. Similarly, one more tRNA produced, and this tRNA take one you see, and that is U, then A, and then what? G, and this AUC. So AUC once again is codon, and for this AUC the codon C, and it is isoleucine. Isoleucine, ILU. So isoleucine. So means once again one more link is formed, and that is called as a tripeptide bond. If it is more than 2 bond, and that is called as what? Here polypeptide bond. So like this, idikku kuda agtade, and here is also there, and here is again again glycine, and here is a proline. Okay, it is taken place like this. If it is a bond is formed, and that form one complete polypeptide bond. Polypeptide polypeptide chain, sorry. So polypeptide chain is formed, and that polypeptide chain means it is here the protein, and at the end what happen? See here, chain initiation complete, chain elongation, chain initiation take place here, and everywhere there is a blocking of what? The ribosome also play very important, because the ribosomes are the protein synthesis factory, and so that also play very important role in move from one region to the another region. When the mRNA diffusion to the cytoplasm, the ribosome come into cover the AUG, and that is again important. Ribosome illandre protein synthesis agodilla, and so thereby the ribosome play very important role in read the frame, read frame, and catalytic reaction also it continue. And in the beginning chain initiation, once the chain initiation by the methionine, and then the chain elongation from the next, to the next, then the next, like this it goes on elongate, and the result in the formation of polypeptide chain. And finally, when the stop codon comes, UAA, UAG, and UGA. So these are the stop codons. So more stop codon is the more. So UAA, UAG, particularly see UAG. UAG is a stop codon, and where there is no any amino acids or anticodon arise for this. This stop codon is also called a non-sense codon. ಇದಕ್ಕೆ ಸೆನ್ಸ್ ಇಲ್ಲ, ಹಾಗಾಗಿ ಇದಕ್ಕೆ ಯಾವುದೇ ಅಮೈನೋ ಆಸಿಡ್ ಏನಾಗೋದಿಲ್ಲ, ಬೈಂಡ್ ಆಗೋದಿಲ್ಲ. ಸೊ ದೇರ್ ಬೈ ದೇರ್ ಇಸ್ ನೋ tRNA to this, and there in this way here the protein synthesis stop, and that process is called as what? Chain termination. And as it the protein synthesis stop, large number of releasing factors also produce. Releasing factors. Large number of releasing factor, number 1, R1, R2, R3 like this, and these releasing factors help in separation of one protein or polypeptide from the mRNA strand. mRNA in the separate ಆಗ್ತದೆ. So in this way, one polypeptide chain or one protein molecule is synthesized, and after the end of the translation process, and this is about the protein synthesis. I think you understood. Very briefly I have explained. So nothing to be make complex ಮಾಡ್ಕೊಳ್ಳೋಂಥದ್ದು ಏನೂ ಇಲ್ಲ. So chain initiation after the first AUG ಅಲ್ಲಿ, and that particularly it codes for methionine amino acid start ಮಾಡಿ ಕೊಡ್ತದೆ. ಅದರ ಜೊತೆಗೆ ribosome play very important. Thereafter ribosome move from one place to another place by continuing the same here polymerization. So this is one methionine is amino acid, then link is taken place to the another arginine. So this the one after the one ಒಂದಕ್ಕೊಂದು amino acid link ಆಗ್ತಾ ಹೋಗೋದಕ್ಕೆ polymerization ಅಂತ ಕರೀತೇವೆ. So that is why polymerization of amino acid, isn't it, into polypeptide. ಎಲ್ಲ ಅಮಿನೋ ಆಸಿಡ್ ಒಂದು ಕಡೆಗೆ ಕೂಡೋದಕ್ಕೆ ಏನಾದಾಗ ಪಾಲಿಮರೈಸೇಷನ್. ಹಾಗೆ ಪಾಲಿಮರೈಸೇಷನ್ ಆಗಿದ್ರೆ ಮಾತ್ರ the polypeptide chain, and that is a protein. And when it ends, and when the termination, particularly UAA, UAG and UGA are the here 3 codon, and these 3 codon stop the process, and where further elongation is not taken place, and thereafter releasing factor here release, and that releasing factor help in releasing the entire protein molecule. So this is about the process of translation you studied. Easy, is there nothing to be complex. Once a book over it, and you can understand this concept. And next one is what? And the next one is the regulation of gene expression. Regulation of gene expression. What is this actually? So regulation, controlling of the gene expression. Actually gene expression is regulated, means the entire process is controlled by four important processes. In this four important process, the levels are there. At every process there is a different level is there, and you must know that entire process of gene expression or the gene regulation that had taken place in the cell only, and that to be in the nucleus and the cytoplasm. See, the four levels are there. First one is transcription level. First one is transcription level. Second one is processing level. Processing level. And next one is transport of mRNA from nucleus to the cytoplasm. Nucleus to the cytoplasm. Okay. And last one is translational level. Translation level. Already all four you studied, and again there is no special here. Transcription. See, transcription, transcription is a process of here what? The formation of RNAs from the DNA, and particularly the mRNA, rRNA and the tRNA that already you studied, and this is a transcription. Once the transcription taken place, processing is taken place. Processing level. What is that processing level? So processing level means transcript, a transcription unit is already immature. ಅದನ್ನು ಏನು ಮಾಡಬೇಕು? ಮ್ಯಾಚುರೇಷನ್ ಮಾಡಬೇಕು. And that maturation process is called as processing level, and wherein the splicing, major the event that is taken place is splicing. What is the splicing? So splicing means arranging of all exons in the sequential or proper order and removing the introns. Then what are the capping and the tail. Iಷ್ಟು process ಆಗಿದ್ರೆ here the mRNA processing taking place, and that to be converted into hnRNA, heterogeneous RNA, and that heterogeneous nuclear RNA. So that is hnRNA, and that hnRNA is a processed mRNA. That one is what? The processed mRNA, that you must remember. So that is why processing is very important. If in the nucleus, after the formation of mRNA, if processing is not taken place, and such do not go but continue for here next processing ಹೋಗೋದಿಲ್ಲ ಅಂದ್ರೆ translation ಗೆ ಹೋಗೋದೇ ಇಲ್ಲ. So the processing is very important. Then once the processing is completed ಅಂದ್ರೆ maturation, mRNA maturation ಆಯ್ತು ಅಂದ್ರೆ, and then transport of mRNA from the nucleus to the cytoplasm. ಈಗ ಎಷ್ಟು ನಾವು ತೋರಿಸಿದೆ, ಈಗ this is a cell and here is the nucleus, and the DNA produce RNA, and this RNA now diffused into cytoplasm, and that is transport of mRNA from nucleus to the cytoplasm. ಅಂದರೆ ಈಗ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಂದ ಸೈಟೋಪ್ಲಾಸಮಿಗೆ RNA ಬರಬೇಕು, and where it diffuse, where the ribosomes are present, particularly in eukaryotic cell 80S ribosome, and the prokaryotic 70S ribosome. But however in prokaryote there is no nucleus, but only in eukaryote there is a nucleus is there, and it 80S ribosomes is important, and to that it attach, and that help in carrying the catalytic reaction of the protein synthesis, as well as reading the mRNA frame, entire mRNA frame ಅನ್ನು read ಮಾಡಲಿಕ್ಕೆ ribosome ಬೇಕೇ ಬೇಕು. ಹಾಗಾಗಿ ribosomes are, that is why called as a site of the protein synthesis, and that is a very important. Transport of mRNA from the nucleus to the cytoplasm is over. Now next one is a translational level. So this translational level you already studied. Translational level in which here the three processes are there. There is one is a chain initiation, correct? Chain initiation, then chain elongation, and the chain termination. All that you studied, and that play here a very important role in completion of the here the second phase of the protein synthesis, that is the translation. If translation complete, and then only we will get the complete gene expression. Complete gene expression. Gene expression ಅಂದರೆ protein synthesis. See, ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಎನಿ ಜೀನ್ ಯಾವುದೇ ಜೀನ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಬೇಕಂತಂದ್ರೆ ಅಲ್ಲಿ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಪ್ರೋಟೀನ್ ತಯಾರಾಗಲಿಕ್ಕೆ ಬೇಕು. ಅಂಡರ್ಸ್ಟುಡ್? either our body character, internal character, or any hormone secretion, enzyme secretion, or any chemical is required. For that all, one or another protein is required, and that protein produce ಆಗಲಿಕ್ಕೆ ಬೇರೆ ಬೇರೆ ಎಂಜೈಮ್ಸ್ ಎಲ್ಲ ಸಿಗಟ್ ಆಗಬೇಕಾಗ್ತದೆ. ಆ ಒಂದು ಪ್ರೊಸೆಸ್ ಏನಂತ ಹೇಳ್ತಾರೆ? ಜೀನ್ ಎಕ್ಸ್ಪ್ರೆಷನ್. ಸಿ, ಈಟನ್ ಫುಡ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಡೈಜೆಸ್ಟ್. ಅದು ಡೈಜೆಸ್ಟ್ ಆಗಬೇಕು ಅಂದರೂ ಕೂಡ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಗಬೇಕು. Because all enzymes are made by the protein. See, like this, for in every our catalytic and metabolic process, here gene expression or protein synthesis is essential, and without which here no metabolic process taking place. If any metabolic process is stuck, so means it is not properly operating, means there is a problem in the expression of the gene, or there is a problem in your food deficiency. Some part of the your component is you are not providing. ಯಾವುದೋ ಒಂದು ಪ್ರೋಟೀನ್ ಅಮೈನೋ ಆಸಿಡ್ ಯಾವುದೋ ಒಂದು ಅಮೈನೋ ಆಸಿಡ್ ಬೇಕು, ಅದನ್ನು ನೀವು ಸರಿಯಾಗಿ ತಿಂತಾ ಇಲ್ಲ. ಯಾಕಂದರೆ ನಾವು ಡೈಲಿ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್, ಪ್ರೋಟೀನ್, ಲಿಪಿಡ್, ಆ್ಯಂಡ್ ಪರ್ಟಿಕ್ಯುಲರ್ಲಿ ಪ್ರೋಟೀನ್ if it is digested, you will get the end product is what? Amino acid, and from that amino acid only in our body again the protein is prepared, and that protein that to be prepared according to the requirement of the cell. ಸೆಲ್ಲಿಗೆ ಯಾವ ಟೈಪ್ ಬೇಕೋ ಆ ಪ್ರೋಟೀನ್ನ ತಯಾರು ಮಾಡಬೇಕು. ಯಾಕೆಂದರೆ ಸೆಲ್ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಮಾಡಬೇಕು, ಜೀನ್ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಆಗಬೇಕು. ಆ್ಯಂಡ್ ದಟ್ ಬಿ ದ ಡಿವಿಷನ್ ಆಫ್ ಸೆಲ್ ಡಿವಿಷನ್ ಆಲ್ರೆಡಿ ಯು ಸ್ಟಡಿಡ್. ಆ್ಯಂಡ್ ಸೆಲ್ ಡಿವಿಷನ್ ಇನ್ ವಿಚ್ ದರ್ ಆರ್ the two main phase. There is one is the M phase, another one is the interphase. Interphase is the most important, and during that interphase only, remember, protein synthesis taking place. ಅಂದರೆ ಅಲ್ಲಿ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸನ್ನು ಮಾಡಿ ನಂತರ ಎರಡೂ ಡಾಟರ್ ಸೆಲ್ಸಿಗೆ ಈಕ್ವಲಿ ಆಗಿ ಅದನ್ನು ಡಿಸ್ಟ್ರಿಬ್ಯೂಟ್ ಮಾಡಬೇಕು. ಸೊ ದಟ್ ಇಸ್ ವೈ this is the most important process, and with this you are going to study. Now next one is lag operon concept. Lag operon. Lag operon. See, what is this lag? So lag means lactose. Lactose, you know, disaccharide, carbohydrate is. Lactose is a disaccharide, it is a carbohydrate, you know. And operon, it is one systematic. Operon and many other scientists, Francisco Jacob Monod, lag operon, Francisco Jacob Monod, who first time discovered this concept, lag operon concept. There are so many different type of concepts are there, and such concepts are used to study the expression of the gene expression. How exactly the gene expression taken place, and what type of expression taken place, and wherein what type of the enzymes are involved, and the repression protein is formed. On all that base, here the lag operon concept base, there are different type of lag operon is there. There is one is called the tryptophan operon. Different type of operon is there. TRP operon means a tryptophan operon. The next one is valine, VAL operon. So many different type of operons are there. Particularly tryptophan ಅಂದ್ರೆ amino acid, VAL ಅಂದ್ರೆ valine. So lag ಅಂದ್ರೆ lactose. Like this, different type of operons are there, and these operon what means is operating condition, on what base they operate. And this was first time explained by Jacob and Monod. Francis Jacob and Monod first time they explained this operon system, and details of all that operon system, and how the process of transcription and then translation taking place, and what is the role of the enzyme, etc. That will continue in the